this computer. All right, let's get started. Are you guys hearing me? Okay, so this afternoon, as our tradition has been unfolding here, I will do the honors of introducing the introducer of the speaker. So today, Alana, you have to tell me how to pronounce your last name.
It's Chatra.
What she said. So I've known her, even though I can't pronounce her last name well at all, since about a year or so ago. Alana was in my chemistry lab class, did a research project with me. She's a major here in biochemistry on a pre-med track, a junior in her third year. She hails from Whitby, Ontario, Canada, and went to Kingsway College. I think we have a number of Kingsway College students that have come our way over the years. Alana hopes to become a physician and a novelist. Yes, she and I have spoken quite a bit over time here about her interest in writing and literature and stuff. So that's pretty cool. I love science. She loves science as well as creative writing. And she plays the piano and the cello. So it's your turn to introduce our speaker today.
All right, thank you so much, Dr. Murray. Today, I have the honor of introducing Professor Christy Haynes, the Distinguished McKnight University Professor at the University of Minnesota. Professor Haynes was born in Arizona and completed her undergraduate work at Macalester College with a major in chemistry and a minor in mathematics and Spanish. She earned her PhD in chemistry at Northwestern University in 2003, and her work focused on plasmonic nanomaterials and applications of surface-enhanced Ramen scattering, a study that is still being continued in her independent laboratory that was launched at the University of Minnesota in 2005. In addition to the title of Distinguished McKnight University Professor, Professor Haynes has also been awarded the Theophilus Redwood Award from the Royal Society of Chemistry and the Craver Award from the Coblenz Society most recently. She has been recognized as an Alfred P. Sloan Fellow, a Searle Scholar, a Dreyfus Teacher Scholar, and a National Institutes of Health New Innovator. Professor Haynes is currently the Associate Head of the Department of Chemistry at the University of Minnesota, the Associate Director at the National Science Foundation funded Center for Sustainable Nanotechnology, and an Associate Editor for the journal Analytical Chemistry. She also leads the Haynes Research Group, a lab dedicated to applying analytical and nanomaterials chemistry in the context of biomedicine, ecology, and toxicology. So please join me in giving a warm welcome to Professor Haynes as she begins today's lecture on polymer-enabled plasmonic sensors.
Oh, that was an amazing introduction, Alana. Thank you. That's all very humbling for sure. Hi to everybody who's online. So I'm excited to visit today. I wish I was visiting in person, although I heard you all were doing virtual everything today for weather purposes anyway. So maybe if I was there, I would still be doing it this way. So I know I only have about 40 minutes, and my goal is just to tell you about one of the projects that's happening in my research lab at the University of Minnesota. My group actually does a wide variety of stuff, as you maybe guessed from Alana's introduction, and I'd certainly be happy to kind of answer questions or emails about any of the other topics that I didn't choose to cover today. And I'll also just mention that I'm also happy to answer questions you might have about kind of graduate school or research experience for undergraduate programs. So with that, I'll kind of jump right in, and I assume somebody will get my attention if you have a question along the way that you want me to try and answer. So Alana gave you the kind of short version of my talk title. The one that you see on the slide right now is the kind of longer one, right? If you read a scientific journal, the titles look more like this. So let me dissect the title a little bit so that you know where we're going. leveraging linear polymer affinity agents and surface-enhanced Raman scattering for the detection of food contaminants. So this is going to be an analytical chemistry kind of sensing seminar. I'm going to try to demonstrate how we detect food contaminants. And you can see we're going to use some polymers, and we're also going to use some nanoscale surfaces to do this technique called surface-enhanced Raman scattering, which I promise I'll define for you. There are two graduate students in my group that contributed to the majority of this work. Dr. now Rebecca Rodriguez, she just defended her PhD in November, and Dr. Victoria Schlag. So I will give them credit right now and again at the end. Okay, so let me kind of jump right in with a little bit of motivation. I told you we're developing sensors to detect food contaminants, and perhaps this is totally obvious to all of you. Every time you hear on the radio or read on social media about a food recall because of listeria or salmonella, we all know that food contaminants are an issue. And in fact, they're going to become more and more of an issue as the world population grows, as global food sustainability becomes more of an issue. Foodborne diseases are going to obviously cause more and more issues. And so right now, about 600 million people worldwide experience some kind of foodborne illness every year. And it's an expensive process. About $100 billion a year is spent treating foodborne illnesses. And as I'm guessing you can imagine, in parts of the world or cities where there are less food resources, foodborne illnesses end up being more and more of an issue based on poor storage or things like that. So food contamination matters. And when we think about what food contaminants might be, you can actually find them in all kinds of categories of species. So there are definitely protein contaminants. Anybody on here who has an allergy to, peanuts or something like that, of course, deals with trying to avoid protein-based allergens. There are also biotoxins like the molecule ricin, which is a bioterror agent that's a protein. There are small molecules. Anybody who cleans their fruit before they eats it, right, is trying to get rid of small molecule pesticides. I'll also talk a lot about mycotoxins today, so you'll hear more about that. Bacteria like salmonella or E. coli, everybody wants to avoid those. You can also have foodborne norovirus or hepatitis. Fungi are obviously something we all want to avoid eating. And then, of course, parasites are an issue. So lots of different types of food contaminants that one might want to try to detect. So how do we detect them? You can imagine if you've taken analytical chemistry or an instrumental analysis class that there are lots of ways to detect food contaminants. And of course, it depends on the target, right? You detect a virus differently than you detect a small molecule. But you will find methods that cover all of these different major pieces of instrumentation. UV vis absorption spectroscopy is a super simple technique. And if you have a molecule that's colored that you're trying to detect, of course, that's a great way to do it. Chromatography is a really common way to analyze complex samples, right? As long as you have a standard and you can see when it comes out of the chromatograph, you can detect things. I have a personal love for electrochemical methods. So if you have something where you can do some redox chemistry, there's lots of, the reason I love electrochemistry is because it's so quantitative. So if you want to know how much of something is, you really can't measure anything smaller than like a single electron. So electrochemistry is a very nice way to do it. I'll talk about vibrational spectroscopy today. It has a lot of advantages, as I'll explain specifically. And then mass spec is a beautiful technique for detecting things, whether you know exactly what you're looking for or you don't. Unfortunately, it tends to be pretty complex to do mass spec and pretty expensive. So when we think about the different methods one might use, we think about the kind of typical figures of merit that you think about in analytical chemistry. So what is the limit of detection or quantification? How hard is it to do? How expensive is it to do? So today, like I mentioned, I'm going to focus on a vibrational spectroscopy. And probably you've heard the most common vibrational spectroscopy people run into is infrared absorption. So maybe you've done some infrared, you put your sample in an FTIR and you look to see where the major vibrational bands are. So vibrational spectroscopy is very nice to work with because, of course, it tells you about the structure of a molecule. And so Raman spectroscopy is complementary to infrared. Actually, the selection rules for determining whether something is going to give you an infrared peak or a Raman peak are complementary to one another. So the general idea for Raman spectroscopy, and sorry, I should clarify, Ramen, not like ramen noodles, Raman named, yes? sorry, Raman is named after the person who first discovered the phenomenon. So C.V. Raman discovered this in 1928. But the general idea is if you shine light, and usually it's laser light, onto a molecule that's vibrating, most of the light that gets scattered away from the molecule is exactly the same wavelength as the light that hit it in the first place. So this is called Rayleigh scattering. But a very small number of photons actually scatter at a slightly different energy, because the molecule, as it's vibrating, can actually absorb some energy. So there's two different types of scattering that are part of Raman scattering. Distinguishing them isn't important today. But what I'll say is Raman scattering is highly unusual. So only about 1 in every 10 to the 8 photons is Raman scattered. All the rest are Rayleigh scattered. So they are very hard to detect because you're looking for a photon that has a slightly different energy from this giant ocean of photons that all have the same energy as the original one. However, it is worth the trouble because, of course, it tells you about the structure of the molecule. It tells you about the functional groups that are there. It tells you about the conformation and the orientation. It's a beautiful technique for detecting molecules and knowing exactly what you have. But like I mentioned, it's inefficient. So really, the way to use Raman spectroscopy is to use a slight variation of Raman spectroscopy called surface-enhanced Raman scattering. So it's the same inelastic scattering process, but now the molecule that you're going to detect needs to be near a very specific kind of surface to enhance that Raman scattering. The surfaces that most people use for SIRS, this brings in a little bit of nanoscience, are nanoscale noble metal surfaces. So make nanoparticles out of something like silver or gold or copper. And the reason that these I'll call these plasmonic substrates, the reason that they're important is because if you can imagine, when you think about a metal, you think of a sea of electrons. If you shine the right wavelength of light on a silver or gold or copper nanoparticle, you can actually cause those electrons to oscillate collectively. And of course, when you have electrons oscillating, what you do is you set up electromagnetic fields. And so if you have a Raman active molecule sitting in those big fields, suddenly the induced dipole gets a lot bigger and you get a much bigger Raman scattering signal. So you can enhance that, kind of feeble Raman scattering by a factor of like 10 to the 8 or even up to 10 to the 12, simply by putting the molecule very close to a nanoscale noble metal surface. So the wavelength of light that makes that collective oscillation happen is called the localized surface plasmon resonance, or LSPR. You may hear me mention that. So essentially, by putting your molecule near these surfaces, you can get to much lower limits of detection, which is important for creating a sensor. So surface-enhanced Raman scattering data looks kind of like the cartoon I have here. This is not real data, it's just sketched. It's generally presented like most other spectra. It's called the Raman shift. It's presented in wave number shift. And on the y-axis is just intensity. And so what you get is very complex spectra. The bands are narrow, and there are a lot of them because every part of the molecule that is vibrating, almost every part, can give you a Raman band. So one way that you can figure out what your Raman spectrum means is you can do a computational calculation about which vibrational modes should give you a Raman band, and you can compare that to your experimental data. Another way that you can interpret the data is actually by using chemometric techniques. And if you haven't run into this, essentially you kind of use the power of linear algebra to kind of sort out what features in the spectra are similar in a group, and you can kind of sort them based on what their spectral characteristics are. And then one last way, you'll see a little bit of analysis this way today, is you can analyze Raman using different spectra, which is exactly what it sounds like. Essentially, you collect 2 spectra, one before your molecule is there and one after, and then you subtract one from the other. And changes in those vibrational bands tell you about what appeared or disappeared from your substrate. So I mention this just because you're going to see some Raman spectra, and it might be a little bit confusing about how we would interpret them, but I wanted to give you a general idea. Okay, so I've told you we're going to do surface-enhanced Raman scattering. I want to tell you quickly the different easy ways one might create those noble metal nanostructures that you need in order to get those electromagnetic fields that give you SIRS. So a very common way people do this is they make noble metal nanoparticles in solution. That's what's depicted on the left-hand side of this slide. It's very easy to do this. Hopefully some of you have had a chance to make some noble metal nanoparticles. You put in a salt like tetrachloroaurate with a reducing agent, and if you have the right reaction conditions and surfactants, you can make some very nice nanoscale colloidal solution particles. That's very useful if you want to do detection in a liquid phase. Often people want to do detection on a solid substrate instead. And so in the middle of the slide, you can see one way people might make this, make a nanostructured substrate. So in this case, put down some polymer nanospheres, some beads, and then deposit metal on top and remove the mask, and whatever reached the surface is left there. Or on the far right-hand side is a substrate that I'm going to focus on today, a really simple SIRS substrate called a metal film over nanosphere. So let me show you all the steps of making these. It's actually pretty easy. You start with a flat substrate. We use silicon, glass, mica, copper. You can use, if you need something conductive, for example, and then we can actually buy a nanosphere suspension. These are kind of hundreds of nanometers, the nanospheres, and you can commonly get them made out of polystyrene or silica. You want the most size monodisperse colloids that you can get because they self-assemble into a close pack lattice. Here you can see a little electron micrograph showing you a top view of what those nanospheres look like. Then you can thermally evaporate metal on top of them. And so this is a cartoon where we've put some gold on top of the nanospheres. And now you can see, if you look closely from this top view, that you don't just have the smooth spheres, you also have the kind of roughness based on the depositing the metal. You can see this even a little bit more clearly if you split the sample and do a cross-sectional electron micrograph. And so here you're seeing these metal film over nanospheres, and you can really see the gold kind of on the top half and the smooth nanospheres on the bottom. At that point, you can do whatever you need to do to build a sensor. And what I'm going to show you today uses some polymers that we put on the surface that are designed to have some affinity for our target. So then you introduce your target, and now hopefully the thing you want to detect is held nice and close to that nanostructured surface so that you'll get a big surface enhanced Raman scattering signal. So that's the goal for making the substrate. The very first time my group did some work with polymer affinity agents is depicted in this little cartoon here. I will say using polymers as affinity agents is pretty unusual. Most commonly, you've probably heard talks where people design sensors where they use antibodies, which of course have very high and very specific affinity for targets. But they're pretty big, which makes them not great for SIRS because you really need the molecule you capture to be close to the gold substrate. Another really common affinity agent is an aptamer, a piece of DNA or RNA that's been selected to bind, again, very tightly to a specific molecule. But my group wanted to detect the molecule ricin, which I mentioned earlier is a protein-based bioterror agent. And we knew that ricin actually, the way it attacks cells in the body is it interacts with sugars on the surface of the cells. And so we decided we would use a glycopolymer, a sugar-based polymer. You can see a little cartoon representation there, and hope that it would grab the ricin so that we could detect it. And in fact, that worked. And so we became really interested in this idea of using polymer affinity agents, in part because you have so much different chemistry you can access with polymers, right? Just by choosing slightly different monomers. And you have lots of control over whether you present a really short polymer or a really long one. So we moved from that little bit of ricin example I showed you to what I'm mostly going to share today, which is the detection of small molecule toxins called mycotoxins. And we chose mycotoxins because they're a really important food contaminant. Essentially, it's estimated that 1/4 of the world's crops have some mycotoxin contamination. Mycotoxin isn't a molecule, right? It's a class of molecules. And you can see all of the crops that are commonly contaminated. And actually, you need to detect this because some mycotoxins can make people really sick. And so you definitely want to make sure you eliminate them before people consume the food that you're dealing with. Sorry, there we go. So there's lots of mycotoxin testing that's already done. It actually has to be done in order to sell food in many global markets. Here you can see just a little graph showing you how much mycotoxin test it has increased over the last 5 years. Well, the last 4, I guess. We're not in 2023. That's a prediction. And the main ways that people test for mycotoxins right now, kind of in the field, are one, using dipstick methods. So you can see in the picture here, if you've ever, lived in a place where there's a pool, right? You often use dipsticks like this to kind of look at a range of chemicals that are in the pool water. In the case of mycotoxins, there are aptamers on each of the different pads to detect different critical mycotoxins. Another common test that's used is an ELISA, which I'm guessing many of you have run into the idea of an ELISA. This is an antibody-based sensing system. Now, these definitely work, but both aptamers and antibodies are really specific affinity agents, meaning you have to isolate a specific aptamer to bind to a specific mycotoxin. It's not ever going to let you detect more than one. So we wanted to try out the polymer approach to see if we could detect not just a specific mycotoxin, but actually more than one with the same affinity agent. We decided to start with the aflatoxin you see on the screen now. You can see the molecule here. This is called Aflatoxin B1. It's a really obvious choice for a first mycotoxin to try because it is the most potent natural carcinogen that's known. So it is definitely something you want to detect if it's in your food. Of course, that means lots of people spend money on making tests for these. I've also shown you on the bottom some of the mean kind of exposure levels in a few places in the world. So these values are in nanograms of aflatoxin per kilogram body weight. So you can see kind of what the exposure levels are. And I've also listed there how the FDA and the EU version of the FDA regulates aflatoxin B1. So in the US, the limit in a foodstuff is that there's 20 parts per billion of aflatoxin B1. And you can see the EU is even more strict. You have to have less than 2 parts per billion. And that's, of course, a safety standard. So we want to detect aflatoxin B1 using surface-enhanced Ramen scattering. And the reason we want to use SIRS is because, of course, we know we'll be able to see the specific fingerprint for aflatoxin B1, and we'll be able to distinguish it from any other mycotoxins or other background things that might be present in complex food samples. So the goal here is, have enough specificity to grab a molecule, use SIRS so you can actually distinguish it, but have the polymer be general enough that hopefully you can detect more than one small molecule target. So how do you choose the polymers? I don't know how much you all have run into polymer chemistry in your coursework so far, but there are a lot of polymers out there. And we decided to start by looking at some computational predictions that had been made. Specifically, there was no sensing here. This was not a Raman paper. It was a chromatographic separation of mycotoxins. And what happened is the theoreticians modeled the 3 polymers you can see listed here, the NN, methylene, bisacrylamide, acrylamide, and allylamine. Those are the monomers. And they modeled how the thermodynamics of interaction between polymers made out of these monomers and aflatoxin B1, as well as another mycotoxin called ochratoxin A. That's the binding energies you can see here. And essentially what we realized is that these would be great potential affinity agents for mycotoxins for our SIRS sensor. However, these 3 monomers are actually not great if you want to do a controlled polymerization, which we do, because we need to keep our polymer relatively short so that when it grabs the target, it's close to that nanostructured surface. So instead, whoops, I didn't mean to switch there. So instead, we made polymers out of the 3 monomers that you can see here. And if you look at them, you can see structurally, they have a lot of similarity to the ones were in this paper. So you can see why we chose them. So the 2-aminoethyl methacrylamide, which we'll call AEMA, the hydroxyethyl methacrylamide, which we'll call HEMA, and the N-acryloyl glycinamide, which we'll call NAGA. So we started with those 3 monomers, and we wanted to make polymers out of those. And when I say we, I should clarify that I am not a polymer chemist, but my 2 graduate students that were on the first slide, Becky and Victoria, both had a close connection to a polymer chemist in my department, Professor Teresa Reinecke. And so they used a technique that you can see listed here, reversible addition fragmentation chain transfer polymerization, also known as RAFT, to make polymers. And I won't get into the gory details here. I'll just say that in general, when you do a RAFT polymerization, you start with a molecule called the charge transfer agent, and that's what you see down here. And essentially, you introduce your monomer, and it's a controlled polymerization. So you can pretty nicely select exactly how many repeats of the monomer you're going to have. And I'll say there was some strategy in choosing this chain transfer agent. If you notice, there's 3 sulfur groups in the chain transfer agent. We knew we wanted this molecule to stick onto gold, and there is a very high affinity between sulfur compounds and gold. And so this was a nice strategic choice to have that at the end of our polymer. Okay, so we made these polymers. the polymer AEMA, HEMA, and NAGA. The first two on here are much easier to make because you can actually buy the monomer. For NAGA, you actually have to make the monomer, which is a little bit more challenging. And we made 4 different chain lengths of each polymer. So we've got a total of 12 polymers here. So the question is, how do we pick which polymer is going to be best? The one way we tried to approach this is using a technique that I'm guessing you haven't run into. I definitely had never heard about this when I was an undergraduate, but it's a really cool technique called Isothermal Titration Calorimetry, or ITC. And the general idea is you mix together your 2 things. So in this case, our polymer and our Aflatoxin B1, and you essentially monitor the thermodynamics. You monitor how much heat is absorbed. And so you're looking at 2 sample traces here where we've measured ITC heat exchange. And on the left is one where we have polymer and aflatoxin. In the middle is a control. So there's no heat of interaction because there's nothing interacting. And then we can essentially plot, as you see on the right, the delta H for each injection and characterize how strong the interaction is between 2 things. So we went through and did this, and sorry, there's so much data on this slide, but what you're looking at are these heat of interaction plots for the 3 different polymers. So the AEMA on the left, the HEMA in the middle, the NAGA on the right, 4 different chain lengths for most of them. The numbers telling you how many monomers are in the chain length. And what's really obvious from this plot is that the shorter chain lengths had a stronger interaction with aflatoxin B1. You can see they've got bigger delta H's, the red and the black lines. And so what we saw, shorter chain lengths seemed to be the way to go. And specifically, we picked out the poly NAGA with 22 repeat units. You can see that's the red line in the far right plot there as targeted for our further studies. So we were essentially using thermodynamics to figure out when there was a really strong interaction, and then we wanted to translate that onto our SIRS substrate. So this is a little complicated, but it's showing you kind of how we were going to allow our polymer to interact with our nanostructured surface and how we were going to allow it to interact with aflatoxin. We wanted to figure out what was the way to get the most sensitive system. So in this picture, the little strings of purple circles are our polymer. So think of that as our 22 repeat NAGA polymer. When it has a little purple triangle on the end, that means it has a trithiocarbonate end group. That's the piece that came from the charge transfer agent that I showed you. And when there's a green teardrop on the end, that means we've converted the trithiocarbonate to a thiol, and I'll explain that in a second. And then if you compare B and D to C and E, the main difference is whether we attach the polymer to the surface and then flow over the aflatoxin, or whether we let the polymer and aflatoxin interact first and then introduce it to the substrate. And I know that sounds a little bit tedious, but when you're trying to figure out how to get the best limit of detection, you have to kind of try out all these different angles on the sensor. So let me show you some actual data now. Before we put any polymers on the surface, we of course wanted to check and make sure we even needed a polymer. So we did the control experiment first, where we just had the gold film over nanosphere, or FON, didn't add any polymer at all, introduced aflatoxin, and looked to see if we could see a SIRS signal. So you're seeing some Raman spectra in the middle here. The black one is a negative control, so there's no aflatoxin there. And then as you go up, you're going to higher and higher concentrations of aflatoxin. And if you look in that little gray bar, you might see that there's like a tiny new peak that shows up, but it is very small. And if we go through and analyze that peak, which is attributable to aflatoxin B1, a specific ring breathing mode, what we see is that we can actually distinguish, 1 parts per million aflatoxin from 0. That's what the statistics tell us. But that is not near good enough, right? 1 part per million is 1,000 parts per billion, and the FDA regulates this at 20 parts per billion. So clearly we need some way to make the signal bigger. And for us, having a polymer grab the aflatoxin and hold it close was obviously the way we were going to try and proceed. So there's a lot of data on this slide. I will orient you. So this is our NAGA polymer with the trithiocarbonate end group, and we stuck it on the surface in advance, and then we flowed the aflatoxin over. And what you're looking at is the 4 different chain lengths of the NAGA. So in black is the 14 mer, in red is the 22 mer, in blue is the 30 mer, and pink is the 38 mer. And what you're seeing in those colored spectra is the polymer only. And then in brown, in all cases, is after we've exposed it to 50 parts per million of our target. And you'll notice in the red and blue spectra that there are some new bands that actually appear when we put in the aflatoxin target. They're highlighted by that gray box. So that was exciting. It seemed like our polymer was actually helping us to detect this. So we proceeded with that 22 repeat NAGA and tried different concentrations of aflatoxin. So when you develop a sensor, you always try and figure out, what is your limit of detection. So again, in black is just the polymer. It's our negative control. And as you go up, you're getting increased concentrations of aflatoxin. And we do see a new band. That band that's highlighted in gray actually seems to be a combination of both some piece of the aflatoxin and the polymer that's there. And when we go through and look at how the intensity changes as a function of the aflatoxin we've included, what we see is that we do better than when we had no polymer. Now we can do 100 parts per billion as our limit of detection. Still not good enough though, right? 20 parts per billion is what we need to get to, or even lower. So we were concerned that trithiocarbonate end group was kind of making it harder to do the detection. When you have more bonds, of course, you have more vibrations, which means you have more Raman modes. And so we were worrying that that was blocking some of what we were looking for. So here you can see that some of the peaks for aflatoxin we're going to be looking at are in this 1200 to 1400 wave number shift region. And that's where a lot of the trithiocarbonate bands are. So we did a chemical reaction and converted our polymer from a trithiocarbonate termination to just a thiol, which is much simpler, still sticks to gold really nicely. And when we took that polymer, I can show you the Raman spectra comparing them. So in green is the usual trithiocarbonate, and in orange is once we've turned the polymer into the thiol. And you'll notice that there are a couple places where there's much less spectral congestion. So we were anxious to try the same thing where we pre-mix our thiol terminated NAGA polymer with aflatoxin and then introduce it to our surface to see if we can get an even better limit of detection than the 100 parts per billion we had before. So that's what this data is. So thiol terminated 22-mer of NAGA interacts with aflatoxin, then we put it on the surface. The black spectrum is the negative control again, so there's no aflatoxin there, and you have increasing amounts of aflatoxin as you go up. And we do see a nice new peak that definitely is attributable to our aflatoxin. And when we go through and actually quantify how big that peak is compared to a control, now we are doing much better. We can actually distinguish, we have a limit of detection of 10 parts per billion. So we were actually able to get below that 20 parts per billion limit, which was really exciting. It told us that we had something that might actually be potentially useful. So some conclusions from this little part before I show you the next part of the research, just to make sure we're all on the same page. These linear polymers do have good potential as affinity agents. There's lots of good reasons to use polymers. Both antibodies and aptamers are biomolecules that require kind of sensitive conditions. And so linear polymers are much more robust in that way. We found that allowing the polymer to interact with the target ahead of putting it on the surface gave us a better limit of detection, and that using the thiol instead of the trithiocarbonate gave us better results. So that propelled us towards the next stage of this study, which was trying to do some multiplexing, meaning we wanted to detect more than one thing at the same time. So imagine you have a food, some kind of food, in this case it looks like orange juice, that has two different mycotoxins in it. And it's really not just the mycotoxins, right? It's everything else that's in that food. So it's particles, it's proteins, it's ions. So if you wanted to detect those two toxins in the usual way, using an ELISA or using those dipsticks, you would need two different antibodies that each have affinity, or two different aptamers that each have affinity. So I think I've been clear here that our goal is to have one polymer that can do more than one thing. So we decided to expand from just aflatoxin to two more mycotoxins. You can see them depicted here. The first is called deoxynivalenol. I'll call it DON for short. It is also known as vomitoxin, so you can imagine how pleasant it is. It's known to contaminate wheat and grain, and actually it's regulated at 1 parts per million, so a lot higher limit than the previous work with aflatoxin. The other toxin we decided to add was Okra toxin A, which sometime you'll see written as OTA. You can see it contaminates grain and pork, and actually Ochratoxin A really makes people feel terrible and you can tell because it's regulated at a much lower level at 5 parts per billion. So if you remember the structure of aflatoxin, you look at the structure of these two, DON and ochratoxin, you'll see there are some similarities, there are some differences, lots of opportunities for hydrogen bonding in both cases. And so we thought that a polymer would be good for looking at all of these. So we were going to proceed kind of as you could guess we would, based on the first bit of this talk, where we were going to take our polymer, allow it to interact with our targets, and capture a Raman spectrum. But now we want the same polymer to interact with multiple targets. So in this case, the little green triangle is meant to be DON, and the little red oval is meant to be ochratoxin. And ideally, we'd get a Raman spectrum that has distinct bands for each of the different mycotoxins that are present. And because then ideally we would know both are there, we might even be able to quantify each of them separately. So that's what we're going for in this work. You might worry that the different vibrational bands or Raman active bands for the two molecules might be too similar, and maybe you wouldn't be able to tell them apart. And in fact, these spectra weren't published. So Becky did some computational work and predicted the vibrational spectra for the two molecules. So the one for DON is in green, the one for ochratoxin is in that kind of reddish color. And you'll see there is some overlap, but there are also some really distinct features. Having her do these calculations was also really helpful because it let us, when we later on captured the spectra, it let us actually understand which parts of the molecule were closest to the surface or interacting with the polymer. So back to the same 3 polymers that I mentioned to you before, we continue to work with these 3 polymers. We screened all of them for affinity with both of those molecules, the DON and the ochratoxin. And we realized that the AEMA polymer, the one that's here on the left, seemed to be the one that had the most potential for multiplex detection here. So I'm just going to show you a few spectra. These ones are bigger, so hopefully you can see them better. The first, I'm just going to show you the polymer that performed best. We did that same selection using isothermal titration, calorimetry, and all the early Raman experiments. We decided that this 29mer seemed to be the one that had the best affinity. And so we put it on the substrate. And so the bottom 2 spectra that you're looking at are negative controls. One is just the polymer, one is the polymer in the solvent conditions that the DON is presented in. And then the top 2 spectra are with 2 different concentrations of the DON. And even if you don't know what Raman spectra are, you can see there are lots of differences between the bottom 2 spectra and the top 2. So there are lots of really nice clear bands that show up when the DON is held close to that surface. When we do the same thing with ochratoxin, again, the bottom 2 are the 2 negative controls, and the 3rd, 4th, and 5th from the bottom are all different concentrations of ochratoxin. And here again, you can see there are bands that both grow and change, and there are new bands that appear when the ochratoxin comes in. So these were both just one and then the other, so just like our aflatoxin experiment, but Becky did what we'd been wanting to do, which is present both toxins at the same time. So the very bottom spectrum you see here in dark red is just our polymer control. So it's that same AEMA 29mer on the bottom. And then as you go up, the next, the greenish kind of spectrum is just the ochratoxin at the federally regulated limit, so 0.005 parts per million. The dark blue one is just the DON, again at the regulated level. And then the kind of aqua colored trace at the top is the 2 of them presented together at the limit. And what you'll see is there's a lot going on here. The green bars that I have there are the peaks that show up that are very clearly due to the DON. The red ones, there's only 2 of them, but they are the ones that show up that are only ochratoxin. And then all those gray bars are features that change in both cases. That's either because they have shared vibrational modes or because it's actually a change in the polymer when it binds to one of the 2 toxins. But either way, lots of very specific bands that we can look at to distinguish that both mycotoxins are there. So we dug in a little bit to try and understand exactly how the polymer was interacting with the targets, because of course we want to think about optimally designing a polymer that can bind to whatever we like. And literature suggests that the ochratoxin can form hydrogen bonds with with the actual monomer unit. And so Becky went through and did calculations, and the band we ended up looking at most to try and quantify okra toxin was this one at 1527 wave number shift. And you can see in the little movie here that this is a vibration happening on the phenylalanine part of this molecule. She did the same analysis for the Dawn. There we were looking at a shift around 1450 wave numbers, and you can see that this was all about movement on a tertiary carbon in the hydroxyl group of the molecule. Quite a big movement there. So we were actually able to kind of sort out exactly where the bands came from, and it made sense in terms of they were parts of the molecule that we know are likely to have hydrogen bonding interactions with the polymer backbone. So I can give you some little conclusions on this multiplexing piece. Obviously, this is still work in progress, but I feel like we're starting to get a real understanding about how the polymers interact with the molecules, which tells us ways to generate maybe even better polymers to use in the future. We've been able to demonstrate that we can detect more than one mycotoxin at the same time. And while I didn't show it here, Becky's actually continued this work and has shown that it doesn't just work in perfect solvent conditions. She's actually been able to spike all of these mycotoxins into cereal, which is, of course, a grain-based product, which is where these mycotoxins end up, and also into beer, again, a grain-based project. And she's been working on doing shorter and shorter incubation times to see how fast we can do detection. And one thing that is really nice about this work is that actually the analysis is very simple, right? If you go back to the spectra, even with your naked eye, you can see that there are spectral differences. And so we don't have to do a lot of really complex data analysis in order to do these detections. So with that, I'll do acknowledgements. I, of course, just lead the group. The really important research progress was made by Dr. Rebecca Rodriguez and Dr. Victoria Schlag. Those are the two students in my group that did SIRS. Dr. Seyoung Jung is the polymer chemist who worked with my collaborator, Professor Teresa Reinecke. And George Schatz is a computational chemist at Northwestern who helped with some of the early modeling with his students, Mark and Antoine. And Sam Bryson was actually a really excellent undergrad who worked with Victoria and Rebecca to make a lot of the substrates. This work was initially supported by 3M, which is actually right in our backyard here. They have a very large food contaminant detection bit, and they really wanted to see if SIRS had potential. It was also funded by the National Science Foundation. And I'll close with just one more slide because I want to mention, based on timing, that this is the perfect time to apply for undergraduate research. If you're interested in traveling to another institution, lots of departments have what we call RRU programs. The University of Minnesota Chemistry Department has one. The one I have on the slide here is part of a center that I help run called the Center for Sustainable Nanotechnology. Alana mentioned that during my intro. This research center actually stretches across 12 different research institutions. So when you apply, you can actually say, oh, I would like to go to Minnesota, but I don't want to go to Madison. Or you can just say, I'm willing to go anywhere, but I want to do cool research related to nanotechnology and sustainability. And our application deadline is coming up. It's a perfect time to apply. We also have a very specific program. If we have anybody on the line who is a veteran, we do some extra professional development for folks that have served our country in that way. So I would encourage you to apply. I'm happy to answer questions either about the science, about University of Minnesota, about the RU program, but I'll stop sharing there. And yeah, I can take questions in the chat, or you can unmute yourself, whatever is easiest for all of you.
Okay, thank you very much. Okay, so we want to encourage all our students, faculty as well, to ask questions, make comments.
I'll just say, I can just add maybe that when I was an undergraduate, I had definitely never heard of Ramen scattering. And so even if you have a question that you think is kind of simple, or maybe you should know based on having just listened, please don't think that. Ask whatever you like. I'm happy to answer anything.
Okay, very good. So I have one question about the attraction of these mycotoxin, these, like the aflatoxin and stuff, to the polymer. So you, I think, said that it's mostly hydrogen bonding. Is that based on just, comparing the functional groups and you know, sort of intuitively saying, or do you actually have some sort of information that tells us it is hydrogen bonded?
Yeah, the reason I say that, I mean, it can be based on intuition, but really what I based it on is, I don't know if you noticed how complex the Raman spectra were, but in every Raman spectrum, we have, you know, the polymer by itself first. And then when we add the aflatoxin, sometimes those polymer bands shift or grow. And so that actually tells us that there's conformational change in the polymer. And so sometimes you can kind of pair, oh, I see this aromatic phenylalanine, you know, character of the mycotoxin show up, and I see the shift in the backbone at the same place. And so it tells us, it's not like we can detect hydrogen bonds, right? But we can detect changes in kind of how the polymer is oriented on the surface that give us good clues that it is a hydrogen bonding interaction, that essentially it's, you know, the polymer is being disrupted by the presence of the newly added analyte. Does that make sense?
Yeah. So have you guys studied different types of polymers with different degrees of either hydrogen bond donors, bond acceptors? Because that could also help in designing of future.
Yeah, you're right. It totally would. And we have incidentally done that, right? Because we've got different polymers, and of course they have different hydrogen bonding ability. We have never intentionally designed polymers to ask that question, right? To say, like, I am building a polymer where everything is as close from polymer to polymer, everything as similar as possible except the hydrogen bonding ability. We haven't done that. You're right. It would be an interesting study, and polymers are a perfect way to do it, right? Because you have more control and more choice in terms of functional units than you do with most other molecules. So it's a good suggestion.
Okay. Anybody has a question, you could unmute and ask a question. You could put it in the chat, but I prefer that you unmute and ask your question. The other question I have is, what type of safety, I don't know, I'm assuming extra safety procedures and protocols do you guys use in all these mycotoxins?
Yeah, you're right. No, your instinct is, I mean, obviously my poor student does not want to be directly exposed to vomitoxin, right? So yeah, in order to have these molecules in the lab, we have to go through an extra biosafety certification. We have to have labs that are actually certified at the right biosafety level. And then on top of that, the students that work with the mycotoxins actually have to go to our environmental health lab, and they get fitted with a really intensive respirator. I mean, we're all used to N95 masks now. This is way beyond that. It has kind of active canisters on it that she has to change out kind of every time she, you know, is done in the lab. So we work very hard to make sure that her respiratory health is not compromised by doing the work.
Wow. Yeah. That's, yes.
I have a question about the disposal.
Yeah, go ahead.
With the disposal of the mycotoxins, like to piggyback off of the working with them. So how is that done? Is it done on campus, or do you guys use as another source, or
Yeah, no, we do it. So anything that comes in contact with the mycotoxins, whether it's that substrate or whether it's the vials where we make dilutions, has to go in a special biohazard kind of containment unit. And then essentially we indicate that to our safety folks. And when they pick it up, they handle it in a different way. It's all incinerated, honestly, at the end, but we want to make sure the people that are doing that waste handling know that there's something different in there. And I'll just say that, you know, we are a giant institution. I don't know if you know how many people are at the University of Minnesota. It is 45,000 people. We are the biggest PhD department. So we generate tons of chemical waste. And I will say this is definitely not even the most dangerous thing that our waste folks have to handle. So we're very careful about it. Yeah. We're very careful about labeling, but yeah, you have to be. Yeah.
Yeah. All right, thank you.
Yeah, of course. Thanks for the question.
I actually want to bring Ryan into this conversation, because I'm thinking if polymers work, your favorite molecule, Ryan, you know what we're talking about? Dendrimers.
The Dendrimers, yeah.
Yeah, have you seen any reports about using Dendrimers as sensors for these food contaminants, these mycotoxin?
Are you asking Ryan or me?
I'm asking you both, actually. I'm asking you both.
Go ahead, Christy.
I have not seen it. I have not looked for Dendrimers in terms of doing food contaminant detection. I've run into lots of Dendrimers when people want to do kind of ion-based detection, or most of what I've run into with Dendrimers are people that want to use them to kind of template nanoparticles. So essentially, you know, plating out ions that are in those tiny cavities. So if Ryan has seen this with food sensors, I'd be curious to know.
I haven't. We're actually working on some PAM-AM Dendrimers as copper sensors. They seem to be really specific to copper. You know, actually, one of my first jobs out of Northwestern was working on a molecular imprinted polymer project. And this reminds me a lot of that. And we didn't have much success. And I think part of it is looking at your target that you're trying to go after. I think we were trying to sense some really big proteins. And also I think your sensing technologies is also very different as well. And I think the Raman helps because you can help sort out some signals.
Right.
And we were just doing like, I'm trying to remember what sensing technology we even used. Even
like probably some electrical sensor or something would be my guess.
It could have been. It was very just specific to whatever kind of mass it was holding on. And so it was so non-specific. It was hard to attribute the capture. It just felt like we were enhancing the surface area, and it was capturing more because we had more surface area rather than a very specific interaction. And so it didn't go very far. So using polymers and trying to design the specificity in there, it's actually quite hard, and why a lot of people do use antibodies and but you guys have, I think you've coupled, I think the Raman helps.
Well, right. So that's the point of Raman, right? The point of Raman is that you get this very specific fingerprint for a molecule. And so you can use a less specific affinity agent because all your specificity comes in the signature. People do the experiments the other way all the time, right? Where you have a more specific affinity agent and then you do like fluorescence detection where you don't get any specificity. So I think either of those work. But if you want to use Raman, I mean, there's no reason to have a super specific affinity agent. Like, why not make it more broadly applicable, I think.
So
I just want to say to some of the students who haven't heard of Raman spectroscopy, come take instrumental analysis, because that's what I'm teaching right now. And we do cover that. So it's really cool. And I always think of Van Dyne's work. And I remember we actually had one of our students do a summer REU at Northwestern. I think he was in Van Dyne's group, and they did, it was 10 years ago or more. And just looking at single molecules, I mean, it's just a really cool technology. And so we do a set of experiments with Raman, but it's not enhanced at all. So we're trying to detect like molar concentrations of something, but
well, that'll give you even more appreciation for what I was trying to do, because Raman is not naturally built for, trace detection, but man, if you can do it, there's a lot of advantages. Right. So and Professor Hayes, who he's referring to as my PhD advisor, was Rick Van Dyne, who is the discoverer of this phenomenon. So that's awesome. Why I'm in space.
So I was wondering if, however, thinking about the potential to use Dendrimers could be limited based on what you're finding, that you're preferring the shorter chain polymers. And isn't it true, Ryan, that most of the Dendrimers out there are like, extended generation?
Different generation Dendrimers, but I mean, my sense of why that wouldn't work is mostly, I think of the openings, if you will, in the Dendrimers as being relatively small. That it would be pretty hard for even like these molecules to find a way to kind of get in. And the reason the length is so important for SIRS, I'm sorry if I didn't say this, is those electromagnetic fields that give you the enhancement, really, they die off exponentially as you leave the surface. And really, you want the thing that you're going to detect to be within like 2 nanometers of the surface. So if you put a big antibody on the surface that holds your target 5 nanometers out, it's as though it's not there. So we really need, like, as little as possible for the affinity agent.
Yeah, the Dendrimer, like if you had a really small generation 0, that may help, but yeah, you're right. Because as soon as you get into a larger one, that's a great point. You'd be outside the
You're outside that
Raman.
That sweet spot, the sampling volume, we'll call it.
Yeah. I just wanted to raise that and see
Yeah.
if there was anything there.
It's looking for business opportunities. We actually sell them. We make them sell Dendrimers here.
Yeah.
I wonder if there are any other student questions, or if anybody wants to ask about REU programs.
Well, let me ask. Have you guys have been having a pretty active REU program, and students can apply from anywhere across the country?
Definitely. The REU program I advertised I will say, has done an exceptional job at continuing to have a good experience, even when COVID made things really crappy. In 2020, we had to go to all virtual. There was a lot of stuff that was still useful. Last summer, I had 2 REU students in my lab that had a full research experience. We were able to make that work. I know how much you all who are on this call right now have missed out on research opportunities because of COVID. And so I would say, if you can find REUs to apply for this summer, like, you absolutely should. It is a critical part of your undergraduate experience that COVID has made really hard. It seemed like there was a student that was trying to speak up. Was that you, Zoe?
Zoe.
Here, unmute yourself.
I don't know how many professors are involved in the program, like how many mentors there were for the REU.
Yeah, so it depends on which program. The one I put the slide up for is an NSF-funded Center for Chemical Innovation. So there are 15 faculty across 12 institutions. So it's not a center in like we're all in one building. It's a really delocalized center, and it's very interdisciplinary. And the students that are in all the different labs, we end up meeting up in person with everybody twice per summer, but then you have your in-lab experience. And so it's a huge variety from like nanoscale material science, analytical chemistry, like chemical biology. It's a big mix of subjects. But even like my own department, the University of Minnesota has an REU program that we call our Lando program. It's named after. And there, we bring in 20-ish REU students each summer, and they get distributed among our 35 faculty. So it's a match, right? Like essentially when you apply to one of those programs, you say what your research interests are, and then we try and figure out how to make sure you're somewhere that's going to be most exciting for you. So one really big advantage to doing one of those REU programs instead of just like randomly going somewhere and doing research is usually there's more like kind of programmatic elements. There'll be like professional development stuff all summer. There'll be extra guidelines for your mentor to make sure they're making a plan. So the REU programs are really nice for making sure you're going to have like a really kind of holistic experience.
So the REU that you have cover mostly college students. Do you have anything for high school seniors, for example?
No, so we have not really focused on that, I would say. Like we've had a couple Project Seed students, but we do have a specific area where we're trying to get more community college students involved in research, since that's hard. But no, we've mostly been focused on undergraduates, I'll say.
And what about international students?
Yeah, so international students can apply, especially with COVID. It has been much more complex to actually make it work to get them over. Like the visa issues are a problem, and I don't know how to predict if they will be this summer. So, but international students that are living in the US, there's not the same issue. You can apply in those cases.
Yeah, okay.
Yeah.
All right, very good, very good.
And like, I'm sorry.
Go ahead, Zoe.
One more question about the program.
Yeah.
I know with most REU programs, they have like at the end of the 10 weeks or how long the program is, they have a symposium. Do you guys have that as well? Okay.
Yeah, we do. We absolutely do. I think it's a really important thing. And some people get so nervous about presenting, but it is the highlight for all of us. Like for the graduate student mentors that actually get to see their undergraduate mentee, like kind of put the pieces together. I mean, that's a really important moment. It also prepares you for when you go back to your home institution. Hopefully you can present a poster or, talk about the program to inspire like the next layer of students to kind of apply. So yeah, there's definitely an end of summer symposium. And you're right, it's 10 weeks. And we cover, a stipend, which is pretty decent, plus like travel and housing and everything. We also include money to kind of send you to a scientific conference in the year after if you want, because the goal, of course, is to like launch undergraduates into, a productive kind of research career. And not everybody decides they want to go to grad school after an REU. That's also still a success, like if it helps you figure out what the right path is for you. So it's also a great way to have a really good recommendation letter for whatever you're doing next, because it's a faculty member that's like not at your home institution that can speak to your skills. So it really helps strengthen your application for whatever, whether it's med school or, grad school or a job. Like, I think the connections, the network, the letter of recommendation is really useful.
So a different topic, what about commercialization of your,
Yeah.
sensors? Are you guys working on that? Is that already in the works?
Yeah, so we have some intellectual property protected around the polymer-based affinity agents for SIRS. Actually, I just got a thing I have to sign for the international patent this week. I think we have 3 patents around that space. I have not pursued actually commercializing them, mainly just a bandwidth limitation issue. I think if I ever had a student who was, that kind of, I want to be an entrepreneur, I want to do a startup, I think it would be a really interesting space to work in. I don't have the time or energy to start a company and do it well. So that's why I have not.
I know since I left grad school, one of the things that some grad schools do now in chemistry and stuff is have students take entrepreneurship classes so that they could, commercialize or whatever their research.
I will say here at Minnesota, there's some really interesting programs. We have an NSF, what they call iCore site, which is essentially trying to teach people about how to take scientific and technological ideas and get them into businesses and how to write business plans and how to apply for grants that bring small businesses and universities together. And a lot of students participate in that. There's also, competitions, learning how to pitch your ideas, things like that. Too many opportunities to even keep track of.
Right.
As far as I can tell.
Right,
Yeah.
right. Yeah. Okay, I think we've had a really nice discussion on a range of topics: science, business, REUs.
Yeah, I'll thank you.
Anybody
I'd like
else?
to thank you, Dr. Murray, for the invitation to come talk to everybody. Again, I wish I could see you all in person. This is such a weird way to interact with people, but
I know.
But I wish you all a great and successful semester. I hope you actually get to be in a lab doing things with your hands.
Yes, yes.
Christy, one of the things I would say too, that this is why I don't like Zoom so much. We here at Andrews, we are usually first or second in the United States in diversity. And that's unusual for a small liberal arts college in Michigan.
Yeah.
And so it is awesome being here. We've got students from all over the world, and they're actually represented here in our department. We just couldn't see them with their cameras off, but so it's a really neat place to have.
That's the thing that's very close to my heart in terms of personal mission, for sure. I mean, I would say probably the value that I lead with in my lab is one of trying to figure out how, as a chemistry community, we create a better sense of belonging, so that like everybody realizes we need everybody from all different backgrounds. We're going to get more scientific ideas, more innovation. We're all going to, high tides rise all ships, right? So that's awesome to hear. I wish I could see that in person.
Thank you for joining us today.
Yeah.
It was great. It was really neat to see.
Thanks so much, everybody. Have a good rest of the day.
Okay, thank you. Take care.
You too.
All right, bye-bye.